to today's NPTEL class on the course Applied Environmental Microbiology. So we are in the week 12 session for this course. This is the last session. So until now we have seen about how microbial application is there in environment related aspects, as well as how microbiology studies and techniques work for us to identify and improvise the techniques. So with this we'll start with the session. I'll just open the screen now. So yes, so these are the course content for week 12, which is bioinformatics, that is conceptualizing biology in terms of molecules and then applying informatic techniques from math, computer science and statistics to understand and organize the information associated with these molecules on a large scale. So this is a brief description about what bioinformatics is. So we know informatics is all about dealing with information and getting good insights from these information. So in order to analyze the information and get out some insights out of it, we use math, computer science and statistics. So with them we try to organize the information and understand them. Why do we use math, computer science and statistics together? Just because we have a lot of information, a high number of information we have. So in order to process them and make them meaningful, we use all this statistical and other mathematical techniques. So when the data is completely based on biological sources, we tend to call it as bioinformatics. So basically we deal with biology, computer science and information technology, and of course math when we talk about bioinformatics. So the first question for today is to differentiate between genomics and metagenomics. Okay, so the primary difference between genomics and metagenomics is that genomic explores the complete genetic information of a single organism, whereas metagenomic explores a mixture of DNA from multiple organisms and entities. So when we talk about genomics, we talk about a single organism, a single entity, and we extract the DNA from that organism and try to analyze the complete set of genes that are present in it. Basically a complete DNA analysis of a single organism. Whereas when we talk about metagenomics, we analyze the complete DNA of different organisms that are present in a sample. It's not for one organism but for multiple organisms that are present in a sample. So metagenomics is a collective term. When we talk about genomics, we analyze the complete set of genes but for one organism, whereas in case of metagenomics, it is we trying to deal with different organisms that are present in a particular site. So the next question is, which of the following is not a commonly used bioinformatics tool for understanding biological data? Okay, so try answering this question. Which of the following is not a commonly used bioinformatics tool for understanding biological data? Okay. Uh, so the answer for this question is Blender. So let's start with the first option, which is Megan. Megan is metagenome analyzer, which is abbreviated as Megan, and this is specially used for analyzing the taxonomic and functional content of metagenomic data. So you have a metagenomic data, and you have to analyze the uh, taxonomic groups that are present from the data that you are having, and the functional analysis of a data. So what it does is it takes a DNA sequence from BLAST, and then it maps to the NCBI taxonomy. So this is how we get the taxonomic information. And also the algorithm that Megan uses is lowest common ancestor algorithm to assign the sequences to the taxa. So you have the the sequence information, and it tries to have a comparison from BLAST and maps it to the taxonomies from NCBI. So for this uses the lowest common ancestor algorithm. When we talk about KIME is quantitative insights into microbial ecology. So here it is used for uh, performing the microbiome analysis from the uh, raw DNA sequencing data. So this is basically a pipeline. So it has multiple steps. This is a pipeline through which we can analyze the microbiome from the sequence data. And since this is a pipeline, it does the analysis of microbial communities, including filtering, picking the OTUs, assigning the taxonomy, and generating the diversity statistics, which is the indices basically. So that is what is KIME used for. And BLAST, we all know it is basic local alignment search tool. So when we have a query sequence, we just input it into BLAST and then check from a very big database whether the given sequence matches with any of the sequence in the data space. So this helps us identify similar sequences or the regions of similar sequences, or also to identify the species or genus or the organism that we basically have. And sometimes through analysis it also helps us to track down evolutionary details. Whereas Blender is not somewhere a bioinformatics tool, it's just a image creation or a 3D creation tool. So this is the summary of whatever I told now. The next question is, how does sequencing depth affect the accuracy of a sequencing run? guesses on what is a sequencing depth? Okay. So here, sequencing depth is the number of times we read a nucleotide. So let's say we have a question like, for example, where a place is, where a shop or something is located, we just want a direction. So if we ask one person, there are chances that the direction that they tell is right or wrong. When we ask about some four people, and let's say three people say one direction, and only one person says something a little different, so you can be sure that whatever the three people said would be right. So for any question, if you're going to increase the number of people that you're going to ask the question, obviously the probability of you getting the right answer is going to increase, right? In a similar way, the sequence depth is a similar concept. Sequencing depth means how many times you are going to read a nucleotide when you are sequencing it. So when the depth is low, you can get errors. Like for example, the first time it says that is T, the second time it says it is CA. So there can be chances of error, and this we call it as noise. Uh, but when we talk about sequencing, this can essentially mean that there is some mutation in this. So when we are going to have more error, we tend to think that there is going to be more mutations in the sequence, which is not true, because this is a result of measurement error, and it's not true mutation, right? And also, that is why the sequencing depth is important. So when you are going to read the same nucleotide more than a couple of times, then you can be sure, okay, this is what is present here. So what happens is, when you are going to initially increase the sequencing depth, accuracy is going to increase first. But at one point, what happens is, you have reached a particular level of accuracy, and you know that, okay, this is what is going to be present there. But even after then, when you are going to increase the accuracy, there are chances of mistakes. At this point, there won't be further increase in accuracy, but there will be a plateau. So that is why the right option here is D, where it says accuracy increases initially with the increase of sequence depth, but eventually reaches a plateau. Just like a microbial growth curve that we'll be having, it cannot be increasing forever. When you initially increase the number of reads or the sequencing depth, your accuracy will increase. Further down, it just decreases, or it goes to a plateau rather than, sorry, it doesn't decrease, it goes to a plateau. So the next, so this is the explanation. So the next question is, what is the advantage of using a microarray or a multiplexed PCR over the traditional PCR methods for detecting microbial communities? polymerase enzyme, the nucleotides, and the most importantly we have one aspect called primers. So in traditional PCR we have a single set of primer which generally targets a specific DNA sequence, but when we have some microbial community, we are going to have varied or diverse species, each with a different DNA sequence. So when we are going to use a traditional PCR, we have to use this method over and over again, and it's practically time and money consuming process. When we talk about a multiplex PCR, it is a different version of PCR where we can simultaneously detect different organisms, just because we can use different set of unique primers. So in the same reaction tube in a traditional PCR you will only have one primer, whereas in a multiplex PCR you will have different primers. So whichever DNA is compatible with whatever primer, they get attached, and then the elongation of the DNA strand happens, and hence you basically amplify different DNA from different microorganisms at the same time. So this is the advantage of using a multiplex PCR. And when we talk about a microarray, it is kind of a molecular hybridization technique. So in a single slide you will have different tiny spots containing uh, specific probes. So when your DNA sample, when you are going to load into this slides, uh, you are going to label it with some fluorescent dye and then load it. This will correspondingly get hybridized to the probe that is present in the slide. And then when you are going to analyze it through a laser scanner, you can see how the spots are glowing, and you can do a high throughput analysis basically. So both multiplex PCR and microarray are high throughput methods in comparison to the traditional PCR method. So this is how a DNA microarray works. You have a healthy cell, you have a pathological cell, and you culture them, isolate the RNA, do reverse transcription, and get the cDNA, complementary DNA, and you perform the fluorescent tagging to it. Now we are loading that sample into a microarray, which is a slide with chips. Sorry, it's a slide with multiple uh, holes, each containing different hybridizing tags, and your DNA goes and attached to a specific region onto the microarray. When you're going to use a laser scanner like this, you can see regions with gray, red, yellow, and green. Typically, where gray here represents there is no cell. The green one, because you have a green tag for a healthy cell, and a pathological cell is going to show you a red color. When a particular portion is present in both the cells, it's going to be yellow in color. So this is how a DNA microarray typically works. And in case of multiplex PCR, you have different primers. So for strain A, strain, like different strains, you have different primers you can use. All other components are going to be the same. The buffer, the Taq polymerase, the nucleotides, all. Only based on the primer, this is going to be amplifying the DNA of multiple organisms at once. And the basic steps of PCR is, first one is denaturation, where the sample DNA strands separate. The double stranded separates into a single strand, and each of the strand gets attached with a primer, and then the elongation happens. So a single DNA is now converted into two different DNAs, and in the power of 2 your DNA gets amplified. So that was basically what is a multiplex PCR and a microarray, and how they are different from a traditional PCR. The next question is, which of the following best describes the genome assembly in bioinformatics? assembly determines the order and orientation of DNA fragments to reconstruct the original sequence of a genome. Gene assembly essentially means the processing of large number of short DNA fragments produced by the uh, sequencing machine, and then putting them back together in their correct order and orientation to reconstruct the original genome. So when you do the sequencing, you are going to have it in fragments and pieces. When you do the genome assembly, all these short fragments, or reads as we call them, are going to be put back together into a genome. So this process is called genome assembly. So it helps us determine the order and orientation of DNA fragments. So the first option says that predicting the function of genes based on their sequence similarities to known genes. So this is about functional annotation, which is not a part of DNA assembly or the genome assembly. Once the genome assembly happens, after that only the functional annotation will take place. The C option is about aligning the RNA sequences to a reference genome to determine differential gene expression. Again, this is a sequence analysis or RNA sequencing analysis, just to study the active or expressed genes. When we talk about the last option, identifying and characterizing mutations, again, this is once the genome assembly is done, then only we do all this comparative analysis and stuff. So that is also not a part of genome assembly. And genome assembly essentially includes determining the order and orientation of DNA fragments so that we can reconstruct the smaller fragments called reads back into a genome. So the typical assembly process and the layers of information that they yield is, first one is clone based assembly with scaffolding and finishing steps, and then shotgun based assembly, and then partial assembly to contigs only. So only when you finally finish, so reads, and when they are partially assembled you call them contigs, and then finally only you have a assembled genome. Hope it was clear. If not, please let me know in the chat box. Next question is, there are two statements. Statement A is, BLAST is used in bioinformatics and molecular biology research to identify homologous sequences and infer functional and evolutionary relationships between them. Statement B is, SINA is optimized for aligning large datasets and provides high quality alignment suitable for downstream analysis such as phylogenetic inference and taxonomic classification. So you have to say which statement is true and which statement is false. Or if both of them are true. So BLAST, which is basic local alignment search tool, It is a bioinformatic tool that we use to find the regions of similarity between sequences that we have. So it helps us identify the homologs and also draw evolutionary relationships. Whereas in case of SINA, which is Silva incremental aligner, it is a tool that is designed for alignment of rRNA. So ideally, we analyze rRNA to build taxonomic relationships and phylogenetic trees. So both these statements are true. The difference is BLAST uses a local alignment, SINA uses a global alignment. And in BLAST you have similarity searching, whereas in SINA it is RNA alignment. So by doing BLAST, your output is going to be the statistical significance, whereas in SINA you will get a alignment as your output. This is a fill in the blanks question. Dash generate alignments between a nucleotide or protein sequence referred to as query and nucleotide or protein sequences within a database referred to as subject sequence. You have to just mention the type of BLAST. Sorry, I just lost the connection. So we'll just resume. So dash generates alignments between a nucleotide or protein sequence referred to as query and nucleotide or protein sequences within a database referred to as subject sequences. The answer is BLAST, Basic Local Alignment Search Tool. The next question is, dash is a web tool for multiple sequence alignment, specifically designed for the multiple alignment of ribosomal RNA genes. The tool is also able to taxonomically classify the sequences. correct answer is Silva incremental aligner, or SINA. It is a tool for multiple sequence alignment specifically designed for the alignment of rRNAs. So this is what we use for taxonomic classification of the sequences. We just saw it a couple of slides back. Okay. So the next question is, in BLAST results, a lower e-value indicates which of the following? Any guess? So e value means expect value. Okay. So the significance of e value in BLAST is that if your e value is low, it means that the probability of uh, the statistical significance or the relatedness between two sequences is the occurrence of this entire thing is more by a random chance. Sorry, it's less by a random chance. When the e value is high, it means that the match between the two sequences could have occurred by some random chance or a coincidence. So that is the significance of e value. So when you have low e value, let's say e value is between 0 to 1, okay, in that case lower e value indicates higher statistical significance, which means the match that we see is not occurring by any random chance or anything. It just occurs due to some real relatedness that is there between the sequence. When the e value is very high, it means that there is some noise because of which you uh, get the result as the two sequences that are aligned are related, whereas in reality they are related only by chance but not by significance. The next question is, which matrix is most appropriate for detecting distant evolutionary relationships? mutation, whereas BLOSSOM stands for block substitution matrix. So block substitution matrix means BLOSSOM, PAM is point accepted mutation, and the number next to this PAM is the evolutionary distance between the organism that you want to study. So for example, when the distance between the organism is very low, which means they are very closely related, then you use a PAM matrix or PAM1. So obviously this is not the appropriate answer, because in the question it states that you have to detect distant evolutionary relationship. When you come to BLOSSOM is a block substitution matrix, and here the number next to BLOSSOM indicates the amount of similarity. So if BLOSSOM, the number that suffixes the BLOSSOM has higher value, then it means the uh, sequences are closely related. When you talk about blossom 62, then it is kind of moderately related. But when you go for some blossom with very high numbers, then only it is closely related. Whereas in PAM, if it is very low number, then the organisms are very uh, closely related. So based on this logic, the answer here is high PAM, PAM 250. And finally, to tell about this identity matrix, it accounts for the exact matches, and it does not account the evolutionary things here, because even if there is one difference, it still accounts for it. So you can't study evolution using identity matrix. So which of the following factors affect the alignment score in a sequence comparison? Obviously you should have ruled out one option by now, so just try to answer this question. So the right answer here is the gap penalties and substitution matrix. So what does your substitution matrix essentially do? It has a specific score for the match and mismatch in the sequence. So if a spot is going to exactly match, it has a positive score, and if it's not going to match, it has a negative score. And this is brought about by your substitution matrices, like what we saw in the previous question, your PAM and BLOSSOM. When you talk about gap penalties, let's say you have a long sequence, but if you're going to insert a gap, then your score is going to improve. Then you introduce that gap in the sequence as well. So this is kind of an indicator where there could have been some insertion or deletion in the sequence, and by inserting you have a penalty for inserting the gap, but still your alignment score increases at the end because you introduce this gap. So these are the two factors that mainly determine the alignment score in your um, sequence comparison, whereas the database size and sequence name, internet speed are not the factors. The next question is, which approach best improves the detection of remote homologs iteratively? Remote homologs, in the sense they are going to be local, okay, and you have to perform the process in iterations. So you can wisely choose which approach can be used. So the right answer here is the PSI blast. So PSI blast stands for uh, sorry position specific iterated blast, okay. So in this case what happens is, you have a initial protein protein sequence search, and based on that a matrix is built. Then the used matrix is used again for sequencing and searching. So this process is done in repeats, and that's why it is called a position specific iterative blast. And this is very special for studying the remote homologs, that is the proteins that share the common evolutionary ancestor, but the sequence similarity is very low. Only specific regions or specific proteins are conserved, but the entire sequence similarity is very low. In that case, you use this position specific iterative blast. First you do a general search, from that you find some remote areas, and then you use that search that you did as the matrix for the second search. Similarly you keep on doing it until you get the required information. This is a position specific iterative blast, and the matrix that you use here is position specific scoring matrix, which is a result of the iterations that you perform. When you talk about FASTA, it is sensitive than the normal blast that we do for low similarity sequences, but there is no iterations that happen in FASTA. Similarly, BLAST N, it is comparing the nucleotide sequences, and it is not for checking the protein homologs. Whereas the cluster W, it is a multiple sequence alignment tool. So here we have the different blasts that we have. In BLAST N, your input is going to be a nucleotide sequence, and the database against which it is going to be checked is again a nucleotide sequence, and the alignment is a nucleotide alignment. Whereas in BLASTX, you are going to submit a nucleotide sequence. The software itself will convert that nucleotide sequence into a protein sequence and then do the search with a protein database. In BLAST P, you will enter a protein sequence. The search is against a protein database again. In TBLAST X, that is another version of BLASTX, where in database also you have only nucleotide sequence and you are going to also input nucleotide sequence, but both these data will be translated to protein data and then the search will be performed. In TBLAST N, it is the reverse of BLASTX, where you insert a protein sequence, input a protein sequence, which is checked against a nucleotide sequence by converting that into a protein sequence. So these are the different types of blast that we generally use. Next is, what is the main advantage of high throughput sequencing over traditional Sanger sequencing? Yes, this is a simple question. I'll just wait for one more minute. So the answer is, it gives us high accuracy and longer reads. So high throughput sequencing essentially means you're going to sequence multiple samples at the same time. So you're going to process a lot of data in the same time, or produce a lot of data in the same time. This increased throughput gives us the ability to measure biological features on a genome wide scale in a shorter time frame. It basically saves your time. The next question is, DASH is a publicly available database that stores DNA and RNA sequence and associated metadata such as sequence annotations and literature references. GenBank is a database that stores DNA and RNA sequence data. So MacVector is a Macintosh or Mac sequence analysis application where you can do sequence editing, primer design, database searching, protein analysis, sequence confirmation, MSA, phylogenetic reconstruction, coding region analysis, gel simulation, and other functions. It is the most intuitive, easy to use program available for sequence analysis. Whereas GenBank is the National Institute of Health Genetic Sequence Database. It has an annotated collection of all the publicly available DNA sequences, and it is a part of International Nucleotide Sequence Database collaboration, which comprises DDBJ and ENA. Uh, these three organizations exchange data on a daily basis, so it's the largest publicly available database. The next question is, DASH is a study of proteins and their interactions with other molecules. It uses various techniques including MS, protein microarray, and to identify and characterize proteins and their function. This is a pretty easy question. The answer here is proteomics. Obviously, when you are going to study about proteins, it's a proteomics. Okay, so read this question carefully. What is the purpose of bioinformatics analysis in metagenomics? first option is to assemble short DNA sequences generated by sequencing into longer contiguous sequences. This is the genome assembly part that we were discussing about, and bioinformatic analysis does that. Second option is to compare the DNA sequences in metagenome to known sequence in databases. This is basically the alignment searches that we perform to identify the taxonomy. The next one is to identify and annotate genes present in the metagenome, which is the functional analysis. And then it is the determination of functional potential of the microbial community represented by the metagenome, where you go for
நாளைக்கு
mapping.
இதாம்மா மெசேஜ் இருந்துச்சு பார்த்தா இன்னைக்கு இருக்கும்.
So the final option is to determine the functional potential of the microbial community represented by the metagenome, which means that you are going to annotate the function or something like a metabolic mapping and identifying the function of the community itself, so that to track its function or behavior in a particular environment. So all of these can be done using bioinformatic analysis and metagenomics. So the next question is, dash is used for taxonomic classification of microorganisms present in samples such as water, soil and air. 16S rRNA is used for taxonomic classification. So 16S rRNA is the RNA component of the 30S subunit of prokaryotes, and uh, it is used to study the phylogeny and taxonomy, and it has by far been the most common housekeeping genetic marker for a number of reasons. So the reasons are: it is present in almost all the bacteria, and it is highly conserved, where the function of the 16S rRNA gene has not changed much over time, and finally it is large enough for informatics purpose. Again, 16S RNA sequencing is a culture free method, so you can study the actual bacterial diversity even for the microorganisms which are difficult to culture. So it is commonly used in identifying the bacteria that are present within a sample up to some genus or species level. So the next question is: name a few DNA databases. So GenBank, DNA database of Japan, TIGR, East, and Microbes are some of the DNA databases. Now we come to protein databases. I'll just wait for a couple of minutes to see if you know some. So some of the protein databases include Swissprot, Protein Identification Resource, which are direct and translated databases like Tremble, GenPept, and PDB. Translated in the sense like you have nucleotide to protein translation in the database. So let's see what are scoring matrices. As we saw already, it is the PAM and the BLOSSOM matrices that we saw. So in PAM, the number next to PAM indicates the similarity, whereas the BLOSSOM, so the lesser the number, you use that matrix for closely related sequences. In BLOSSOM, the higher the number itself represents a similarity only. So higher the number, you use it for closely related organisms. And some of the local alignment algorithms that we use are Smith-Waterman algorithm, basic local alignment search tool or the BLAST, and the fast alignment or the FASTA. Okay, so the next question that we have is, which statement best justifies the use of amino acid sequences over nucleotide sequences for distant homology detection? So for you have to perform a distant homology detection, but you are using amino acid sequences instead of nucleotide sequences. Why would you do that? Okay, so the first option is amino acids mutate faster than nucleotides. Second is genetic code redundancy reduces noise in protein comparison. And the third is uh, DNA alignment is computationally slower. And the final option is protein databases are smaller. So the right answer here is the genetic code redundancy reduces noise in the uh, protein comparison. Redundancy means you have different codons that can code for the same amino acid. So let's say DNA sequences have mutations at some silent sites, as we call them. Even though there is mutation in the triplet codon, still that amino acid can be produced because uh, two amino acids code for a single amino acid. Two codons code for a single amino acid. So in that case, this is called a redundancy. So how noise reduction happens is, when you talk about some distant homology, there can be multiple substitutions that are happening that over time. So comparing amino acids like ignores this changes. So only for that you have to compare the nucleotide sequences. So because you have only less number of nucleotides, the random chance of error is reduced and you get more accuracy in your result. Okay, so I'll wait for a couple of minutes for you to finish this question. Just try filling in the blanks. Okay, so FASTA is sensitive than BLAST for DNA comparison. Sequence similarity is quantified by using substitution matrices like PAM and BLOSSOM, and Smith-Waterman algorithm is based on dynamic programming. So in this option B, we know we have already seen. The others you can just have this. The next question is assertion reason question, where Lower E value always means longer alignment, and the reason is longer sequences always give better scores. So here the assertion is false. Lower E value doesn't mean longer alignment, but just that it has happened by some chance. But longer sequences always gives better scores. That also is false, because we saw about how this substitution matrices and gap penalties are involved in the scoring. So the last question for today is, PAM and BLOSSOM matrices are interchangeable. Both measure evolutionary substitution probabilities. the assertion and reason both are false here. They are not interchangeable and they don't measure the evolutionary substitution probabilities. So, with that, we end the session for today. If you have any doubts, please post in the chat box, or else thank you for joining in.